Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share.
Hi, Assalamualaikum warahmatullahi wabarakatuh. So in this video, we're going to continue with subtopic 2.4, which is the last subtopic in chapter 2. So in chapter 2.4, you're going to learn about cell transport. So actually, you already have the prerequisite knowledge about this subtopic, okay, because you learned this during Form 4. So as you know, cell transport, it has two types, the passive transport and also the active transport. So in passive transport, the definition is the diffusion of a substance across biological membrane down its concentration gradient without using energy. So again, the definition is diffusion or the movement of a substance across a biological membrane down its concentration gradient without using energy. So from the definition, the principle in passive transport, first, it is not required energy. And then the movement of substance is down its concentration gradient, mean from high concentration gradient to low concentration gradient. And then for passive transport, it has three types: simple diffusion, facilitated diffusion, and also osmosis. Okay. So for simple diffusion, the definition is a movement of the substance from higher concentration region to a lower concentration region. Movement of the substance, okay, and this substance is referred to small polar molecule, okay, for example, gas, okay, and certain ion from high concentration region to a lower concentration region. So for simple diffusion, okay, it does not require any carrier protein or channel protein because the substance able to cross the phospholipid by layer directly, okay, because they are small and then they are polar. Okay, next, for facilitated diffusion, okay, the movement of the substance from higher concentration region to lower concentration region with the help of transport protein. So the word facilitated mean berbantu. So in this case, the substance will move from higher concentration region to low concentration region, not require energy, but with help of the transport protein. Example is channel protein and also carrier protein, but still not required energy. Okay, this is referred to large molecule, polar. Okay, for example is glucose. Okay, next is the osmosis. It's referred to the movement of water molecule. We must be specific because osmosis is referred to movement of water molecule. Okay, and then for the definition, movement of the water molecule from higher water potential region to low water potential region, okay, across selective permeable membrane. Okay, or you can say that osmosis is the movement of water molecule across a selective permeable membrane down water potential gradient. Okay, kita tak gunakan perkataan concentration gradient, kita gunakan water potential gradient. Okay, nanti saya akan jelaskan. Okay, so from this diagram, okay, take a look at this diagram. So it show, okay, passive transport. Okay, passive transport. This is show a simple diffusion. Why simple diffusion? First, take a look at the first principle. The movement of substance is from higher concentration region, kawasan yang berkepekatan tinggi, in term of substance, bahannya dari kawasan yang berkepekatan tinggi kepada kawasan yang berkepekatan rendah dan tidak memerlukan apa-apa bantuan. Dia can cross directly. Dia boleh cross direct the phospholipid bilayer. And these two is referred to facilitated diffusion. Why facilitated diffusion? First, the principle is the same. The movement of substance is from high concentration region to low concentration region. But then in the diagram, you can see that the movement of the substance is facilitated by the transport protein. Okay, this is referred to the channel protein, and this is referred to the carrier protein. But in the diagram, not sure the using of energy requirement of the ATP. So here, still in passive transport. Okay, but then from this diagram, we can see that the movement of the substance is against concentration gradient. At this side, kita dah tengok ada kepekatan yang tinggi berbanding di kawasan yang atas. Jadi pergerakan movement of the substance ini berlawanan arah dari kawasan yang kurang berkepekatan ke kawasan yang berkepekatan lebih tinggi. And of course, it required energy in form of ATP. Okay, so in active So before we discuss about the active transport, this is you should know about the component of water potential, because when we explain or describe the osmosis, it's referred to the movement of water molecule across selective permeable membrane down water potential gradient. So what is water potential? Okay, water potential is equal to solute potential plus pressure potential. So this is formula that refer to water potential. Okay, water potential equal to solute potential plus pressure potential. So dia menggambarkan okay keadaan ataupun kepekatan air itu, di mana jika water potential air itu adalah tinggi, okay, dia menunjukkan water potential tinggi adalah water potential yang paling tinggi adalah pada bacaannya kosong. Okey, kosong, water potential equal to zero, dia menunjukkan bahawa dalam air itu tak ada, tiada solut. So dengan kehadiran solut, water potential akan menjadi lebih negatif. Okey, sebab bacaan solut potential akan memberi bacaan yang negatif. Okay. So sekiranya kita dapat water potential equal to negative 3 kPa ataupun water potential equal to 0 kPa, so 0 adalah lebih tinggi berbanding negative 3. Jadi pergerakan air adalah daripada water potential kosong, water akan bergerak ke negative 3 punya region. Okay. And then kita tengok pula in terms of water potential, okay, the solution can be divided into three, which is hypotonic with low concentration of solute. Maksudnya dalam hypotonic solution, solutnya adalah rendah. Okay, tidak pekat. Isotonic, maksudnya dia ada same concentration between the water and also the solute. Means the solution have the same concentration, and then the hypertonic concentration is the solution with high concentration of solute. Okay, with high concentration of solute. So dalam kes ini kita nak tengok dia punya water potential. Sekiranya water potential dia adalah negatif, ada tiga situation, katakanlah water potentialnya negatif 3, negatif kosong, negatif 3 dan kosong. So negatif 3 is refer to hypertonic, and then kosong itu adalah hypotonic. Isotonic adalah apabila dia mempunyai kepekatan yang sama dengan surrounding solution. Okay, nanti kita akan discuss soalan ini di perbincangan ini dalam detail, okay, bila kita jawab pasir. Okay, cell condition after immerse in the different types of solution. So we have two types of cell here: the animal cell without. So we know that animal cell is without the plant, the cell wall, and then the plant cell, which the cell have the cell wall. So if animal cell immerse in hypotonic solution, so the hypotonic solution means the solution has low solute. So the water potential in hypotonic solution is high compared to the cell. So movement of water, okay, in terms of osmosis concept, the water from the solution will enter the cell, okay, because the cell has low water potential compared to the solution. So when the water enter the cell will swell and finally burst. Why the cell can burst? Because in animal cell there is no cell wall. Okay, so this condition we call as lysis. Okay. Compared to the plant cell, okay, when the plant cell immerse in hypotonic solution, water from the hypotonic solution will enter the cell, will diffuse into the cell, and then the cell will become turgid, which the cytoplasm and the vacuole enlarge, and dia akan rapat tegang ke arah cell wall. Tetapi since the plant cell have the cell wall, so the plant cell will not burst. Dia tak boleh burst sebab the cell wall is a rigid structure. Okay, so this condition we call as turgid. Next, if the animal cell is immersed in isotonic solution, means the concentration of the solution and concentration of the cell is equal. So water will move in and move out in the same dalam keadaan yang sama, balance. So apa yang berlaku? The condition of the animal cell we call as normal. The condition of the plant cell immersed in isotonic condition we call as the cell is flaccid. Okay, flaccid. So it is different between the turgid and flaccid. So look at the size of the central vacuole and the size of the cytoplasm, which in this turgid condition, dia betul-betul akan sel tu swell. Okay, dia akan mengembang dan betul-betul dia punya cytoplasm akan rapat sangat-sangat pada sel wall compared tu pada keadaan yang flaccid. And then in hypertonic solution, when animal cell immersed in hypertonic solution, so the water potential in the cell is higher compared to the solution. So water will move from high water potential to low water potential. So what is the consequence? The cell will lose water. So the cell will shrink. This condition we call as shrift. Okay, shrift. But for plant cell, the condition is called as plasmolysis. Please remember the term and you can able to differentiate the condition based on the diagram provided. Let's continue with the second types of the cell transport, which is the active transport. So active transport, they have different concept or different principle compared to passive transport. The definition of active transport is the movement of a substance across the cell membrane with use of energy against its concentration gradient and mediated by specific transport protein. Okay, again, movement of the substance across membrane with use of energy against concentration gradient and mediated by specific transport protein. So basically, the principle of active transport, the movement of substance use energy, required energy. Second, it is against concentration gradient, mean the substance will flow from low concentration gradient to high concentration gradient, dari kawasan yang berkepekatan rendah ke kawasan yang berkepekatan tinggi. Okay, so this active transport can be divided into two. First is sodium potassium pump, okay, and second type is the bulk transport. And sodium potassium pump, okay, the concept it show that the potassium ion, okay, is carried from outside into the cell. So the concentration of the potassium is higher inside the cell compared to outside the cell. Okay. And for sodium ion is carried from inside of cell to the outside of the cell. Inside the cell has low concentration of sodium ion compared to outside. So energy is required. Okay. For this, we're going to learn the detail during coordination chapter semester 2. Okay. In bulk transport, we have two types. The endocytosis, which is the process of transporting material into a cell. And the second one is exocytosis, the process that transports substance out of the cell. So endocytosis, kita bawa masuk bahan luar ke dalam sel, dan exocytosis adalah proses untuk bawa keluar bahan daripada sel keluar. Endo masuk, bring into the cell. Exocytosis bring out of the cell. Okay. So for endocytosis, okay, it can be divided into two according to types of the material or substance. First is the phagocytosis. Okay, phagocytosis is defined as a process whereby cell engulf large solid particle. In phagocytosis, it refers to the process whereby the cell engulf large solid particle. For pinocytosis, the process whereby the cell takes in liquid. So as I said, they have divided into two based on the types of the material. For phagocytosis, mean the substance that move into the cell in form of solid particle. But in pinocytosis, the substance that enter the cell is in liquid form. Alright, now let's take a look mechanism during phagocytosis. So phagocytosis is defined a process whereby a cell engulf large solid particle. So let's zoom the diagram so that we know what will happen in specific. Okay. So the first thing here, the engulfing cell detect the particle. So this is engulfing cell will detect the particle. So here the particle is a solid particle. So it's referred to the food. Then the particle will attach to the membrane of of a phagocytic cell. So this is referred to phagocytic cell. The entire cell is referred to phagocytic cell. So the food will attach to the membrane of phagocytic cell, and then the membrane of the cell will invaginate and form pseudopodia. Okay, the membrane of cell will invaginate and form pseudopodia. And then the pseudopodia will surround the particle. So here the pseudopodia will surround the particle, and then the cell will engulf the particle. Okay, the cell then engulf the particle. So this will produce a structure called food vacuole, or in other term also known as phagosome. Okay, phagosome. Next, this food vacuole, okay, after it formed, it will pinch off and move to the cytoplasm. Okay, it will pinch off and move to the cytoplasm. At the cytoplasm, the phagosome or the food vacuole will fuse. Okay, fuse with the lysosome. So as you have learned, the lysosome contain hydrolytic enzyme. So the structure, the fusing between the food vacuole and also the lysosome we call as secondary lysosome. Okay, or in some book it's so phagolysosome. Okay, during this formation of during the fusion of the food vacuole and the lysosome, then it will allow digestion of the food particle. Okay, the digestion of the food particle, then the products will absorb, then the vacuole will disappear, and then just content will secrete out of the cell to process exocytosis. Okay, so here the mechanism: the cell will form pseudopodia, engulf the food particle, and then the cell membrane will pinch off and form a structure of food vacuole. Then the food vacuole will fuse with lysosome that contain hydrolytic enzyme, so the food will be digested. Okay, and then food will be absorbed by the cell. The undigested food or undigested substance will secrete out of the cell by process called exocytosis, and then the vacuole will be disappear. Okay. During phagocytosis, okay, the process is whereby the cell takes in liquid. So here is referred to liquid. Okay, liquid substance. So as you can see here, the membrane of cell, okay, the membrane of cell, it will surround the liquid droplet, and then it may form a structure called phagocytic vesicle. It may form a structure called phagocytic vesicle. Okay, then the liquid content of the vesicle is slowly transferred to the cytosol, or then it will directly absorbed by the cell. So here, phagocytosis involve formation of vacuole. In phagocytosis involve phagocytic vacuole. Thank you for watching. See you in the next video. Bye.